G'day, good morning, good evening. Thank you for listening. This is an organic farming show focusing on plant science, soil science, and microbiology. Information is for educational and scientific purposes and is fact checked. Any of the screenshots, or not screenshots, but papers or websites that have been discussed in today's show will be put in the description below. And if you're watching this back, I haven't got a title for today yet, so I don't know how things are going to pan out. So if there is a show, because last week's was a rad show, so I'm still buzzing off that. So if you want to watch today's show, and if it happens, they'll fast forward along the bottom with your slider thing, and you'll see the screen change when we've hopefully got to some slides. And I'll give another 60 seconds for cool folk to arrive, or even just for the folk to arrive. They don't have to be blimmin' cool, because it might be hot where they are. So if you're listening to this, there's 40 more seconds to go, and I don't have a topic for today. So if you've got anything with plant science, soil science, and microbiology related, please whack in chat, even a discussion topic or a myth. I like the myths, because I can open up a lot of slides, my university slides, and shed light on the topic. They're fun. So another 15 seconds. I have to start talking about something. All right, 5 seconds. And g'day to Genetic Memory Farm says, good evening, good peeps. I hope you all are safe and happy. He says, I am walking the dog, so typing will be messy. Oh well, better than no typing. And it says to me, have you heard of weasels or something, because screw venomous snakes? Pet weasels. Ah, yes, because where I am, it's in snake season in full effect. So he saw a video and suggested weasels. I don't know about weasels here, but you just got to live with them, I think. In Australia, it's just hard. Keep the grass low, keep the brush down, that type of stuff. They've got this snake mesh you can get with small holes. I put it around my chicken coop. But if you're watching this, if you're listening, please put some suggestions in chat, because I don't know what to talk about today. Last week's show was awesome. Wow. Got right into it. It was so enjoyable. It was a good topic we had. Organic versus nutrient inputs. A debate. Yeah, I really like that. So it was pretty cool. Genetic Memory Farms says, I really like my green beans this year and let a bunch go to seed. How long should I let them go? When do I harvest them for seed crop? Like dry and rattling in the pod? Oh, well, what can I show you? Actually, I'll leave that up. Let's have a look at some slides on germination. There's actually one screen I can think of. Oh no. Uh-oh. This one. Hope this is all right. Okay, open when you're ready. So development of seed quality. This is for a different cultivar. So I go through their seed formations: germination fresh, germination dried. That's the blue timeline to the red timeline. and the black timeline is the potential longevity. This says the optimum time to collect them, and over here is the risk of viable loss. So in general, once it's growing, you'll see it growing, and then the plant will start and die off about here, and it'll start and everything will be aging. See just up the top line, dispersal, post-harvest aging or repair, and then the seed's starting to go into the risk of viable loss. Well, when I'm doing mass seed production, a few risks of viable loss is, this could be reworded actually, because it should be, the percents go down very marginally with the days afterwards. It depends on your field conditions. If you've got the risk of viable loss, yes, is huge. If you've got moisture around, because the moisture's got to germinate all the fungus and bacteria, and they're going to start and break stuff down. But if it's really dry, risk of viable loss is extremely low in my experience. So when I'm doing beans, I did some nice purple ones last year, and they weren't all rattling in the pod, because when they dried up, the pods were around the actual, I'm doing this description, you can't even see me. Jesus. I don't want to put my face up, but it's a good way to explain. Where's, no. Here it is. G'day. When the screen now, it's all whack. That's what we're up to. With the pods, actually, I planted it. Some do rattle, but some, the pod on the outside, it'll dry around the seed, and then it'll trap it inside like that. So that's not a good indication for me anymore for the rattling. So the aging, it was shriveled up. It was all shriveled and looking aged. You could easily snap it, so you know the moisture was really low, and the plant had finished. Actually, some of the plant was still growing, and I was still harvesting the pods, but the pods were really dried up. I just waited till they really dried up. That's the point that I wanted to describe, to show them how encapsulated it actually was around it, because it wasn't sitting like the pod wasn't dry there and it's rattling inside. It was actually on top of it. I hope that helps. And that's for that bit. I feel weird now. You don't always want to see me, eh? You don't really care, do you? Good. And back to, when do I harvest them for a seed crop? So last year I did a mass, I can show you that too. All right. Because I've got to put them, I'm throwing them out in the field, which is I made a mass lot of didn't even write it because it's a bit. I got many bags of these buckwheat seeds. See how they're nice and uniform colour as well. All really good. So for me, this is a good example, like I said about the beans, but even for this, I just let it age and really dry till I could strip it off, and it would go, and it would come off like really easily, because it was pretty hard to harvest a few 5 or 6 pound of these, many of these bags. So that's what I did with that plant. Oh, there's a few. I'll put it back in the fridge. You always have to store your seed after you harvest. You've got to store it pretty blooming good. So I know that this is very dry. if it wasn't super dry, I could put paper towel in just to make sure any extra moisture is going to go into it, because you don't want this to start and grow fungus and spoil this whole crop seed. So I'll put it back in. Keep it on low temperature. It's at 3 degrees Celsius, probably 40 F. How long should I let them go? I hope that explains that. When do I harvest them for a seed crop, like dry and rattling in the pod? Oh, next question. Sweet. Yeah, my bean plants, GMF says, yeah, my bean plants are still green and putting out new beans, but the pods are drying. Rad. Perfect. I will try some properly tomorrow. No, let him go. Don't be in a rush, mate. There's a chance you're going to get immature genetics. Let it go, if anything. What's the worst case scenario? If it goes a little bit over, it's not going to. That's what I was up to. I remember now. There's another question. A statement. Nice. I love big wheat noodles. I'm still sharing. Why is the screen whack? It doesn't say sharing screen on the top, but it looks like it is sharing screen. All right, I'll go share screen. Ah, okay. Now it was sharing screen. I'll just press the cross off button. A few issues today. What was up to again? Oh, that's right, being in a rush. I remember now. Share screen. So we're up to this. So the risk of viability loss. He says, I want to be in a rusk. I want to try and get in a second planting for the season. Yeah, I wouldn't. Or I would wait and try and just push it and get a second planting for the season if you want. The viability loss and more moisture. When the pods are really dry, I don't have any in the pods. I took mine out of the pods. Yeah, I did. I got some lovely red kidney beans. Yum. So I'd let them really age a lot, because the risk of germination problems might happen. So for the sake of leaving them on the plant an extra, well, until they're shriveled, so an extra 5 to 7 days might pay off for when you're going to test the germination percentage. And on my germination percentage with that buckwheat is very high. All my percentages are really high. I'd say in the mid-90s, no worries. And then, yeah, so this could be reworded. So risk of viability loss with high moisture. Yes, that's what should be added to this statement here. Yeah, a few extra days in dry conditions, big deal. All right, and if it was going to rain then, what would I do? Oh, well, I'd go big deal, because the seed pods should be encapsulated and not allow water to go inside. And if they're cracked enough to allow water to go inside, I'd harvest them. Yeah, so you can do some selective harvesting on ones that were very, very aged. If it was going to, and then they might. Yeah, yep. I stand by that statement. Buckwheat noodles. Genetic Memory Farm says, okay, I will let them go. That is probably the best question. What is the downside of letting them mature longer? Oh, good question. Hello, Montana. GCC, been a minute. Yes. Hope you're doing pretty good there. Getting ready for the lovely snowfall. The downside to letting seeds mature longer on the plant. All right. Let's have a look, see if I've got any things like that. We'll go back under that seed section. So seed aging on the plant. Seed dormancy techniques? No. I don't know if any of these pictures are relevant. Here's a pathway, a genetic, a metabolic pathway, probably. No, it's not. Seed purity analysis. This might work for you. So you've got inert matter, yuck. Weed seed you don't want, other crop seeds, and you want the pure seed. So you've got to get all the rot out. You already know that. This is a bit off your topic of your question. Your question was, what's the downside to letting them mature longer? They will enter dormancy. If they sit on the plant too long, they'll just sit there and end up not being in the conditions that they like. So they'll enter dormancy. So they've got little live embryos inside of them. That's what I want to show you. It's a bit more. Microbial inheritance in plants. That's pretty cool. That's a pretty cool looking slide. I don't remember this one. Plants to seed, dormancy, and seed to handler. So plant to seed. Oops, endophytes. Oh, the microbes inside, yeah. Okay. Yes, what's this? Interactions with environment microbes, including those associated with frugivorous animals, influenced by post-harvest treatment, storage, and production practices. Oh, there's a bit of a summary. Distinct migration from roots, from seed to seedling. That's when you want to plant it. Okay. So the problem is, this is applicable, I suppose, because you can enter some of these stages where it's in dormancy, where it's just not going to germinate at the percentage that you would like. And imagine a premature baby, human baby, that goes and pulls a bit early. They've got to put it in an incubator and keep it going a bit longer to keep those processes happening for it to mature, for it to have a successful lifespan. So it's a similar type of a thing. It's just not quite ready. Oh, if it was pulled early. And then once it goes into dormancy, the dormancy starts, and then it's harder to break it out of dormancy. You've got to use different techniques to be more successful. So your germination rate would be lower because you've got to use those different techniques. Also, you might have, if you leave the seed on the crop longer, you can enter microbial, specifically fungal problems. Imagine if it's sitting there in there and it's wet. All of a sudden, fungus likes to grow, so it's going to stay wet, and it's just going to go, you beauty, look at all this starch inside, 70% starch. I'm just going to try and attack that food source. And it's over. Yeah, fungi can reproduce in minutes, where the plant cells are hours. Let's see if there's anything else. No. Here's the pathways I was looking for. This is a dormancy pathway. Ambient environment, dormancy induction. So if it was to go into maturity, it would go down this pathway. So it would start and increase the abscisic acid, and it would degrade the gibberellic acid. So it's entering, so in dormancy it'd be hard. That's why using gibberellic acid is very good to bring it out of dormancy. Yeah, I'm not talking about dormancy breaking. It's always a problem, that little bit. See that little black bit? Chink, just pull it off. Always gets stuck. No, oh, here we go. Oh, once the radicals come out, these are the pathways. Ethylene, evolution. That's why using ethylene in your seed germinations is an asset. It goes down to the endosperm. to the CT1 receptor, EFR receptors, response weakening genes, and it happens. Very good. Vertical transmission of seed microbiomes and seed biopriming in maintaining plant fitness and ecosystems. That's another asset. While we're talking on seeds, to bioprime means soak them in beneficial microbes. It's one of the best ways to do it. I'll be doing a big experiment with my garlic next year. So same with your bean seeds. After you crack them, make up a solution and put them in, soak them for, what am I going to soak them for? They say you want to soak them for like overnight and then dry them and then use them within 24 hours. So what that does is the cells that are on the outside, or the spores more so, that have been attracted and attached to the seed tester, the outside seed coat, they will dry and be sitting there. So once it goes into the soil, those little microbes will be sitting there. Once more water goes into it, bang, they start to germinate, which is the same time the root comes out. So that's why it works so blimmin' well. So well. And you don't have to use much either. That's the best bit about it, because you know how much seeds you've got. So you think, that's not many. So you got to make up a little putty solution, which is in a teaspoon even, or in a little tiny container, depends how much you got. So for my buckwheat, I could do it in a little little ice cream container, little bottom of, I got these little milk jug things. I hope that looks like I've missed a few. Oh, I rambled on for a bit there, sorry. It's good to get on with that. Oh yeah, up to there. Hey, there's American one. G'day, mate. Hope you're doing pretty good. I wrote you into last week's show description. Said some thanks. Mainly mushrooms. Hey, mainly mushrooms. How you going? Cheers, bro. Harvest soon here. Woo! Got a lot of fire. Oh, that sounds excellent. Love to see some pics. Weed Nerds is still open. Genetic Memory Farm says, I used to hate green beans until I grew them. Now I love them. I'm okay with dormancy because it may be a season or two until I can run all the green beans again. As long as you store them right, I can get many, many years out of those seeds. Microphile, mesos, and macros, they're the three seed sizes. And you're supposed to get 12 months, 3 years, 5 years out of them. And I can get triple that, no problem at all, if they're stored correctly. And try purple ones, mate. I'm liking the purples. I got, I think, 3 or 4 varieties, and kidney beans. There's my 4th. I've got 4 varieties, cultivars of beans. Hello, Dead Horse. Nice to see you, mate. And so back onto this. I could talk about some more seeds. Do you have any more seed questions? Excuse me. Sorry. Got standard. Dead Horse, eh? I used to live in Whitehorse. And Dead Horse is in next territory over in Canada. Back to seeds, I suppose. What do you reckon? Sort of started on it. How many questions for seeds? Everybody? Seed biopriming process and its impact on seed vigor and plant health. So does it help? Well, this slide suggests it does. It's Trends in Plant Science this is from. So beneficial microbes, inoculation and hydration, There you go, that's how you do it. So you put your microbes into the solution with your seeds, and they're in the solution. Leave it in that for overnight, pull it out. They say let it dry because it sticks a bit better to them. And, well, sorry, the reason why it's dry is because when you disperse or broadcast your seeds, you don't want to broadcast them in clumps. So that's why they have to dry again. And I say use it within 24 hours, but depends how you dry. Your drying conditions, they might be very humid. So just do your best, keeping in mind that those microbes might germinate in the water. Incubation. Benefits. That's where it goes to 12 hours overnight thing. Just got to establish, germinate, improve seed vigor. There'll be all sorts of everything. Depends what microbes you actually use. Try and get ones, be specific. There's many out there now. I use many different types. Put in beneficial bacteria in your search engine. Try and purchase them in the billions of colony forming units, the CFUs, with a B for Bob, because then you've guaranteed that you're going to get some action from that actual microbe because its concentration level is so high. Seed epiphytes and endophytes. What's the blooming difference? Well, endo means inside, and epi means on the surface. So some microbes work inside and some work on the outsides. The ones that work inside are the reason why you need a phytosanitation certificate to import seeds or export seeds now, because they've got live living bacteria or fungi or microbes inside of the seeds that have been transported across the borders. Who's to know what they are, if they're good or bad? That's why people that grow in organics, you'll definitely get that type of effect, whereas there's people that grow in sterile conditions with no microbes, you could sort of say, you won't get this. Their seeds will be very clean and very vulnerable to attacks also too. So by inoculating in your cells, you've taken care of that. And especially instances, if you get your seeds from people that don't grow in organics or use microbes. I'd say 3/4 of people would now though, because people are starting to learn about the benefits of microbes. And they'll have at least some types. I'm finding out of mine, I'm lucky to be I'd say 15% of my microbes are endophytes, quite a low amount. So most of them you'll get will be epiphytes on the outsides. Novel framework for seed microbiome identification and developing effective microbiome tools for plant health. Oh, geez. So seed coding with prebiotics. Oh, there you go. Genomic seed coding with probiotics, single strain or synthetic communities. Seed coding with microbes and protecting substances like nanoparticles. Oh yeah, remember I did that course, nanotechnology in agriculture. That was awesome. Awesome course. Beneficial microbes reintroduction. Yeah, why should you keep them and put them in? Inoculated seed microbes. Yeah, why does people say, why do you have to put them back in? Well, the reason is these roots grow. So if this grows right out to here, sitting out here now, there's no microbes associated, the ones that you want around it, unless they've been vectored across by earthworms or something of the like. But in general, they're not there. So that's the reason why you re-inoculate. Also, if you might get a dominant species, for instance, like Pythium, which is really bad, Umycetes, that you don't want, and you might want to be getting rid of that. So you might pump on the trichoderma and really just Pseudomonas fluorescens and stuff like that, just to really drown it out to try and create a dominant species from the one that was a predator dominant species that you had as the example Pythium. I'll get rattled on a fair bit here. I should see if there's any questions or stuff. Oh yeah, there's a few. Cheers, g'day. GMF says, my other seed crop this year is basil. Oh, that has yet to finish. Should I leave them dry in pods or extract them? Oh, I don't know about basil. Uh-oh. I've never worked with basil. Don't know, mate. I'd say it's exactly the same thing. It'd be smaller. So you've got to be more, what do you call it? More watchful, because beans are huge. These basil seeds would fall in the micropyle size, where the green beans would be in the middle one, which is the mesos, because the macros, which are the largest, would be like apricots, stuff like that are very large. So yeah, you just got to be in the seed size, the micropyle size, you just got to be a bit more extra careful. So should I leave them dry in the pods or extract them? Well, I'd let them dry. I'll just watch the plant. Wait for the plant to go through its senescence, and then you know, right, it's pretty much wrapping up the plant, and then you could do a full harvest of the plant and know, or individual branches, do it on individual branches instead of the plant, possibly, because they're pretty long and stringy, aren't they? Like, that was another question. GMF says they are super tiny. Oh yeah, so in the pod is kind of easier and to keep track and handle. Oh, nice. I'm trying. I want to do clover this year. I want to make my clover seeds, and I'm finding that's going to be hard too. But something new to harvest it by hand. We still talk about, so let's talk about seed biopriming. That's pretty much what those slides were on. Next one. All right. The global seed vault. So that's cool, where it's stored in Sweden, in the mountain. Svalbard Global Seed Vault. 150 meters deep in the mountain, 450 feet in the mountain. Oh, here we go, another one. Vertical transmission of seed microbiome, seed biopriming, plant fitness. The vertical transmissions. Transmission of seed microbiomes across generations, implications for ecosystem functions and legacy effects. Too many big words there, it's hurting my mouth. It's just saying they can get in the environment and help. That's the reason why I use selective beneficial microbes, because I know that they're going to get into my environment and help. Here's an example of bad ones. Like the Indian minor bird, introduced in 1860 in Australia, causing havoc to 3/4 of Australians. The cane toad, introduced in the 1800s, similar thing. Didn't fix the problem, and now causing problems. But these are helping. So the microbes, the beneficial ones, do other tasks too. Then they'll lay dormant until they're needed, and then they'll germinate, create spores, and then just sit there and reproduce that, and then repeat that cycle. So by you sourcing nitrogen, fixing bacteria, they're going to sit out wherever you are in your substrate and reproduce themselves. Getting a bit off the topic here. Oh, these are just explanations of what those slides were before. Okay. It's dormancy. Induction cellular hydrolysis. Okay. Basil and broccoli and beet radish seed are apparently quite valuable as microgreens. Okay. Yeah, okay. I personally like pea shoots for microgreens. If you let your basil go, I'll show you. You want to see some basil that's gone a bit, it goes yellow. Do you want to have a field excursion? Do you want to see the largest carrot in the world? What do you reckon? What do you think? All right. So we're going to do some, you want to see the broccoli. And I'll show you the cross that I did last year, the brock sprouts. No. Some seeds. I put the screen on. I acknowledge that. Before I get out. What else do I need to do? So we're going out to have a look at some broccoli that's gone into flower, and you can all see all these magical yellow flowers. There it is. So you see all those yellow flowers? That's broccoli. Or you can say it's broccoli. That's what your broccoli will look like. That's specifically broccoli. Is this yellow stuff right here. That tall one is a cauliflower plant that was too big for me to eat. And this is all, look, I've got white flowers here. This is from my cauliflower that I bred the season before that. So what they're going to do is they're going to, with your broccoli, you said you're going to let them go to seed. They'll start to form seed pods like this. And then they'll get fattened up. And then you'll see the seeds. How you know on the inside is if they're working, they'll go round. And then you'll start and feel them. And then they'll start and dry up. And then you'll start and flick some of them, and some will start and rattle. And then that'll give you an indication that the seeds are separated from the actual plant itself, so that the seeds are ready. That's an indication for that. Look at the layers. You're just loving life, aren't you? I saw that car stop. Three questions after I show you this big massive carrot. See, these are the brock sprouts that have gone to flower. It's like a humming-bee. You can see the pods there that they've started to produce themselves. What's happening on top of the plant? These are the brock sprouts. They produce little... I have to be careful because I'm allergic to bees, and there's an absolute ton of bees around. See the carrot? It's bigger than my goat. What else am I going to show you? That's about it. These are all the Brock sprouts too, but they've got a lot of bees on them, so I don't like going near these plants. Like stacks of bees. It's humming. It's hard to see. I tried to do a video on it, and they didn't come out in the video, the noise. Yeah, right now. Bee Central. Wish I had a hive. And Brussels sprouts, they're starting to go to flower as well. G'day, chooks. Say hello. There's a rad. Extremely, insanely healthy garlic crop. Really, really, really healthy. This technique that I've done for the first season worked really well. And this is poor-ish land. pH was low. I used a lot of synthetic pesticides here. What else am I going to show you? Oh, there's carrots. Oh, just how they're starting to produce the flower pods. See now in there? That's the flower. So they're going to start to produce a stack of seed. And I'm not going to keep that one, but I'm going to keep this one. Oi, what are you doing? Look at her. Look at the big tongue. Yeah, you do it right in front of me, hey? Look at it, the food's better from this side of the fence. Look at the big massive tongues they got. Oh, I gotta love them. Yeah, this here, see, there's another pod. They go real brown, and then I store them, and then shake them off. Then you got to go and get all of the impurities out of it. Look at this big thing. It's still growing. Yeah, I'm going to get seeds from this one. That's just, it's still growing because that was 12 centimeters. Now it's bigger. That's only 5 inches. Look, there's my hand. Talk about, it's like 8 fingers. Everything. These are the microbes that I use with gibberellic acid in them. There's a little hint for you. Source microbes with gibberellic acid if you want swollen fruit. Swollen. Look at the size of that garlic. That wasn't half that big last year. I'm trying to work with specific endophytes also that have gibberellic acid in it. So I've got ones on the outside and the inside. So the ones on the outside would be down in the soil, producing it for the roots. Ones on the inside would be doing it in just cellular processes where it's needed. Oh, look at that, it's brown. Oh, that's sloppy. A couple of them. Jesus. It's looking pretty good, but. And even that one up the end there, I was going to harvest a few shows ago when we did another field excursion. It's germinated. Can you believe that? Oh, look at that clover's stretched massively. Oh, look at it. That's coming through. I should make sure this is actually working. Oh yeah, I can try. I'll try and read your comments. Personal pea shoots, oh yeah, microgreens. The American one says, I harvested some seed from an outdoor crop a couple of years back. The beans look like mature and the plants were mature, but the seed didn't germinate. Why? Good question. So the seed was mature and the beans were mature. That's, I'd say it's got to do with genetics. It might have been, like, was there a big stress up near the end? There wasn't, because everything was mature. But you can still have stresses that go through without maturity. it's got to be to do with the genetics. When they went through recombination, when the male and the female bits are going through recombination, they go through and then they make their chromosomes go and they cross well and everything's in check. But they might have had some that weren't in check and didn't go through recombination and they mismatched, but that would form a lot of mutants. I would suggest to use some really strong dormancy techniques to break them out, using ethylene from an apple, putting the seeds on top of the water and an apple in the bag, seal the bag up and leave it in the right conditions in room temperature, sort of, 75 Fahrenheit for 3 to 5 days minimum, and then it should germinate then. And if it doesn't, you'd have to go into more harder techniques, maybe cell culture techniques. So you make a cell culture and break the genetics up. So you've got some of the DNA in there that you'd be trying to reproduce that and then get that to grow. So some of the cells that would be inside of them, you get those cells to multiply out of that to form a callus, and then you can give them some descriptions on, all right, you're going to form roots now, or you're going to form shoots from cytokinins or from auxin. But why would it happen? I'd say it's a genetic thing, because if the plant was mature and the seeds were mature, what else can it be? It can be genetics or stresses. And how the stresses just would have coded it differently, but it still would have been gone through recombination. So the things that would be genetic, it's a genetic fault, I reckon. Or you could have been working with triploid genetics, sets of 3 chromosomes, and it could have put it under the conditions that it was needed to. These are shown by bees, not flies. You could put it under the conditions. I can't remember where I was going now, that bloody bee. I'm yellow too. He's had some more comments. I haven't got my glasses with me. Next one. I suppose they have only seemed mature. I'm sure you won't. That could be a possibility. That just because the outside of the coat is the tester stripes on the outside was really nice and dark, that's an indication of maturity. It's gone through the processes to get to that stage, and leading up to that stage, everything's formed, everything's gone well like the plant should, but it couldn't germinate. I'll still come back to, I think it's a genetic fault. Test it, try it. If you bred it, try it again. Try it with a different parent. So if you were putting it with, you were using A was the problem with B, try A again with C, and then you'll prove to see if A is still the problem. So you'll do it with another genetics that actually works well and prove which one won't work, and then see if it is a genetic flaw. That's how you can test it. That's a good question. You reckon it's GMFA? Yes, I reckon it was too. I love those tech questions. I like getting to think, because it's plant science, so it comes back down to the process and how the cells work in the plants. And if you understand that, you just bring it up a bit from there, and what's going to happen next, and what's going to happen next. That's how I was trying to get to the conclusion of that question. I love it. Makes me blimmin'. Makes me think good. Brassica flowers. Yes, that's what's blimmin' humming around me. Again, and I shouldn't be sitting right next to one, should I? Good. This is where I'm sitting. Look at them. They're thick. These are just on these flowers just here. Now look at how many flowers there are. There's a blooming field over here too. Isn't it, Cookie? Yes, you haven't had your leaf today, have you? Not many leaves left. This little way. There's all the potatoes coming up. Some pineapples. Corn's coming up. Over there, heaps of corn's going to be soon. This is where all my beans are. Here's some beans. Oh, they're germinating nicely too. These are purple ones. Oh, look at this. See how it's stuck in the ground? Do you know what type of root that's called? You won't guess what type of root. There's tap roots. There's adventitious roots. There's another type of root called stolon. S-T-O-L-E-N. There it is. It's when a root goes back into the ground like that. Hey, look, it's already getting some fruit on it. When the root goes back into the ground and starts again. So I reckon one of these ends wants to come out. They're both in good. It's going to die. There we go. Oh, look at that. Oh, that's the seed end. I think that was wrong. Is that the cotyledons? That looks like a seed to me. It's on the other end then. Just watch me wreck a plant. No, that was right. That end's pretty in. We'll soon see. We'll see. See if that opens up. Someone needs to be weeding in here too. But yeah, these are all the different types of beans that I got, and I put them specifically next to this bit of mesh. So I just put a star picket in and then put the mesh onto it, and at least that way it can trellis up it. I did some, these are some kidney beans I did last season. They haven't germinated yet. I've got some peas here too. Peas are germinated. There's peas. These things here. Good. Everything's starting to come up. Very good. Oh, look at this type of spinach. This is a silver beet. Got a few varieties. Green. I like this stuff. Like, look how thick the leaves are. That's packed with nutrition. Absolutely packed. Oh, that's gone to flower. See how it's standing up like that? That's going to produce a little flower top. Just like this one. It's a semi-purple variety. My purple patches over there. See all that purple? That's where I produce all my seed. But that whole purple there, that's all silver beet. Different varieties. Again, everything's going really well. So that's the clover. I was going to try and make seed out of. I don't know how to. So it's something I'm going to do this season over there. Not in here, because this is going to be a garlic crop next year. Yeah, so I've got to give you I realise that. All right. I should read this. I said over here there's no bees. American one says, yeah, it was a grill grow, and I had a motorcycle accident. Oh, sorry to hear that, mate. So I didn't go back. And that's no good. So they went to the, oh yeah. There could be a, okay, that's a different, I just think it could just be, it's got to be a genetics thing. If they're not sprouting, try it under the dormancy breaking conditions with the apple. Try it under that. And if that doesn't work, I reckon it's a genetic thing. And if you had if it was a multi-grow, like if you had many species out there, it might be hard. But if you had one or two in particular, you can narrow them down to see if their genetics was weak to start with. I love that question, mate. Good. Keep those hard questions coming. Thank you. I can do that. I'll try different parents. Yeah, cool. Nice one. Because you might have some. You might have a triploid too. You know those like the what's that one that's only available by cutting? Because it's hard to work with the genetics there. Triploids are mostly infertile. Mostly. I'm able to work with one under a stress event. So I put it under stress and then it's reproduced. Up until that time, you can't stress it to get any action. Any thoughts about doing another collaboration episode like last week? Says GMF. Yeah, I'm always happy to do. Let's go back inside. Yeah, that was an excellent show, wasn't it? I really enjoyed that. I'm more happy to do others, get others up here like yourself, like even American one. He's a nice fella, got good banter, polite type. I'm happy to have things up like that. I said to Loki as well afterwards that more than welcome, because he was respectful and didn't cut off. I like to finish my sentence. I talk in pictures, so once I get cut off, the picture goes. It's hard for me to finish. But yeah, any suggestions? I'm more than open to it, because at the moment I don't have anything for next week. There we go. Don't need to see the blue sky. Go back inside. Back in the air conditioning. Yeah, if anybody's got any suggestions on to do different shows or to have collaborations, I don't mind that, as long as the people are positive, nice people. They're moving in a direction that we all want to move in. Just because some grow synthetically, it's they can't help it. They're just in a different situation. What do you mean remove? Oh, I see. There we go. Yeah, so to answer that question, yeah, for sure. Anything in the pipeline or anything organised or stuff like that. But I'd like to. If you know anybody, I don't mind. And they had, he's got a YouTube channel too that's quite popular, and he knows about how YouTube's policies are, so he can talk in the way to keep everything happy in the YouTube community too. I think that was about it. Is there anything more about that was a rad question, the American one. I really appreciate that, about the technical side. Why wouldn't it work? So with a lot of this science stuff, it's about ruling things in and out. So by you doing a few of those little experiments, you can rule things in and out. Then we can go from there and go, all right, next, we'll try this next. And then one might work. You might think, oh, I did manage to, this happened. And then bang, you can think, oh, cool. It's it happened because of the genetics in this one. Or it was did have 3 sets of chromosomes after all. Yeah. This is a good chart. Actually, I should pull up dormancy. I should look at that. Morphology of flowering plants. So the reasons for seed dormancy is a few over here. Natural methods of breaking the dormancy is in the middle, and artificial methods to break the seed dormancy is on the right. So pause this screen if you wish to read some of these. I'm not going to go through them because we'll spend way too long. I've already been doing an hour now. So while we're on seeds, might as well throw this in. There's another, I'll throw in here too, some other slides. These are just half a dozen. Seed of dormancy. I'm only interested in, thank you. Where was that from? University of Kashmir. Oh, okay. 5 different types of dormancy: physiological, morphological, morphophysical, and physical. 7 types: physical plus physiological, chemical and mechanical. Micro seeds, dwarf seeds, they're saying. So I knew about the 3: micro, meso, and macro size. There's a dwarf as well. Cool. See, well, we have slides. Warm stratification, chemical dormancy. Cameron's para-ascorbic acid, ammonia even, ferulic acid, and abscisic acid. Really? What abscisic acid? Oh, because they're dormancy. Yeah, it's ABA. I'm thinking, what? Yeah, inhibitors. Deep physiological dormancy. Cold stratification. Yeah, you're keeping those seeds in the fridge too, Teo? Innate dormancy and forced dormancy. Seed germination. Oh, yeah, it's 3 things it needs to germinate: moisture, temperature, and oxygen. Seeds resume physiological functions and become active. Okay. Grown in the dark, etiolated seedlings. See how they got no chlorophyll yet? It's pretty cool. So seeds can germinate in the dark. They get their, instead of producing chlorophyll or photosynthesis and doing their pathways through that, they'll do it off their starch contents with the night dark cycle. Water imbibition, the uptake of water. Temperature between 16 and 24. Well, no, keep it like a baby. A baby will whinge at 16. 16's blimmin', what's that? 58 Fahrenheit. 24 Celsius is like 70. So keep it like about 70, 75 Fahrenheit in my eyes. You should have, that's what you have dialed in on your temperature gauge. Right, okay, that's it. Oh, you're kidding. Was that not bloody sharing? Did you see those slides then? Hang on, oh, you can see that. Oh, yeah, you saw them. It doesn't say end share button, but. You did see all those slides what we just come up against, hey? I hope you did. Yeah, you can see that. That's right, you can. All right. The share, something weird's happened, so I can't press the, usually in the middle of the screen has a stop share button. It's down the bottom now. And oh, that's not what I was looking at. Yes, saw the slides. Okay, good stuff. All right, well, that's today's show. Oh, hang on. It's a question mark. Oh, I answered that. Oh, good. Sweet. Yeah, even you, I'm happy GMF to come up and answer questions just like Loki did last week, just to sit in the background and just to throw questions in that might keep the topic interesting. Yeah. So you're welcome to that too, mate. Now, I know you don't like showing yourself, so you're welcome to not show your face if you were to do that, but you're welcome. Oh, GMS says, thanks for sharing all your plant knowledge, brother. No worries, mate. I enjoy it. Still way more to come too. With next semester, I've got my exams. I'm in week 12 now, last week of it. Woohoo! And I shouldn't fail anything of the 5 subjects I've done this semester. So that's good. Again, I'll tell you what, that Science of Happiness and Wellbeing course, we might go through that at some stage too, because that really, really helps. There's some good techniques just to bring in happiness, be a nicer person, meditate more, try and unlock the stresses in your mind as well as your body. It's cool, but that's not what the stream's about, but it helped me so much, that course, that I might even throw some stuff in. There you go. GMS says, I could potentially write a couple of loose scripts for your episode. No, just cruisy. It's all just go off the top. If we don't have a show this week, it doesn't matter. There'll always be one next week. I really don't mind. It tells me when the show's has no topic that everybody's happy. And that kind of keeps me happy. I get down after it's 2 or 3 shows in a row. It goes like that. I get a bit sad thinking, oh no, I need to do something. But so, yeah, be easier. Don't know what that means. Do an episode on that wellness course. Hell yeah. It's wait to see what I get in the exam. But I've got 87% so far every week. But yeah, it's helped me a lot in my life already, the little bit that I've done, and a few problems that people would have, PTSD, other things. It makes good suggestions on how that happens, why it happens, and how to get over it. And I'm not going to talk about those things. I'm not a counsellor or a psychologist or anything like that, but just explain the good points on how people might be able to adopt those techniques to move forward. It's cool. Meditation, it actually helps me. The brain flows like as if anyone's good at skateboarding, or they do a manual, and it's sort of, you feel that flow. It's near in the brain. It's odd and very relaxing and calming and nice. All right, not talking about that today. Maybe in the future. I think you'd like seeing the face. All right. Well, it was good seeing everybody again. I look forward to next week's, and I don't know what the topic is. So save all your cool stuff for then. Your plant science, soil science, and microbiology stuff. This time next week, or it's an hour ago next week. 70 minutes. Good show. Good show, everybody. Thanks for the questions. I love them. Keep them coming. Happy breeding. Happy growing. Good health to you all. Bye-bye. You can see me do that for once. I usually sit here and just wave in front of a no camera. See ya. Press that if you like it. Bye.